Hey guys, my name is Will Price, and today we're going to be taking a better look into how statins inhibit fatty acid synthase. Okay, first things first, I'm going to talk about fatty acid synthase. To start off, it has 7 active sites that are separated. They are within a polypeptide chain, and it forms palmitoyl-CoA as its final product. This all forms a whole enzyme complex. This complex is responsible for the synthesis of fatty acids. And right here in this picture I got, which I obtained from class slides, we have acetyl-CoA. And this acetyl-CoA is going to be converted to malonyl-CoA, which is going to be actually pretty important later down in the slides that we'll get into. But from right here is where we're going to start the elongation process, and eventually it's going to form this product here, and this is going to be a repeated cycle until it forms palmitoyl-CoA, and this is going to be hydrolyzed through thioesterase, and it's going to hydrolyze that linkage between palmitate and ACP. And I want to talk about ACP as well. It's right here in the middle, as you can see, and this has a phosphopantethene arm that ends with a thiol. Now, this will become really important later on, and I just wanted to show where some of this is going to be happening to get an image in your head later on down the line when we start talking about these things. So, statins. These are a class of drugs that are prescribed for the prevention of cardiovascular disease with pleiotropic cellular effects. Research into statin effects have become widely relevant, and what you'll learn is that they inhibit HMG-CoA reductase and the cholesterol biosynthesis pathway. They also affect LDL receptor production and lead to the reduction in blood cholesterol levels. And statins also exhibit many cellular effects that you'll come to find out, and that is going to be really the purpose of this article when we get to it. And this whole thing to the side here, we did talk about this in class, and this is really just a way of how cholesterol is going to be taken in. As you can see, we have the LDL particle here and LDL receptor, which is what you see right here is where it's being synthesized and how it gets there, and how it's being taken in, and eventually it's going to get to where it's going to be taken up by the ER. And I just really wanted to show this picture here to basically show you what the statins are going to be affecting in this, I guess, like cellular effects when we're talking about it. Okay, so for the study, they wanted to find out how statins exhibit cellular effects beyond HMG-CoA reductase. The acylation of lysine residues on proteins can impact the function of a modified protein, and before it was thought that this could only occur via enzymatic transfer, but a non-enzymatic method of acylation has been discovered. So this led to them predicting that statin inhibition of HMG-CR would lead to HMG-CoA levels increasing and an increase in protein HMG elation. And as you can see here, I put this picture in to show you where the statins is going to be affecting HMG-CoA reductase, and how where it's going to be inhibiting, and as well as the stuff it's going to cause, as you can see with the LDL right here, and the cholesterol, and the risk with the cardiovascular disease, just so you can get a picture of what's going on here. Okay, so to start, they tested to see if HMG-CoA increased during statin treatment. To do this, they treated hep G2 cells with simvastatin, and which is one of the statins that they're using, and acyl-CoA species were analyzed using mass spec. And out of all the species that they measured, only HMG-CoA levels increased. and this really showed a potent increase in HMG-CoA reductase's substrate. So next, they wanted to see if increases in HMG-CoA resulted in protein HMG elation. And so they incubated HepG2 cells with increasing concentrations of simvastatin across several time points, and while using an antibody recognizing HMG lysine. And what this did is it revealed a singular band of HMG elation occurring at all time points and concentrations. Now, this suggested HMG elation is an effect of HMG-CoA reductase inhibition, and this modification was shown to appear rapidly based on the band of HMG elation being present at the earliest time point. So, interestingly, lysine acylation, Western blots, and proteomic experiments tend to reveal tens to hundreds of modified proteins. And that right there is what they really use to identify the singular band that we talked about. And but this time, like I said, it only showed that one singular band. So they performed a follow-up to discover this protein by performing an immunoprecipitation on statin-treated cell lysate using anti-HMG lysine antibody followed by mass spec. And this revealed fatty acid synthase as the protein, which suggested a link between fatty acid and cholesterol biosynthesis. And so they repeated this experiment in mice models, and it revealed the same results, which suggests an increase in HMG-CoA induces HMG elation of fatty acid synthase due to statins. And over here, I got my picture of fatty acid synthase. This is really a model of it, as you can see, and I really just wanted to put it here so you could look at the structure of it. So, did the modified fatty acid synthase originate from the cholesterol biosynthesis pathway? Well, that's what they wanted to find out. This time, they used a pharmacological tool called hemagglutinin to inhibit HMG-CoA synthase 1. And remember, this is the enzyme that produces HMG-CoA. So this prevented the increase in HMG-CoA levels, which indicate the majority of HMG-CoA detected by mass spec is being used by HMG-CoA reductase. The hemagglutinin treatment impacted HMG elation of fatty acid synthase, so it prevents HMG-CoA increase and prevents HMG elation of fatty acid synthase. So this indicates the connection to the cholesterol biosynthesis pathway, which is what they were trying to find out. Okay, so next they wanted to observe this modification further. So they incubated purified protein with HMG-CoA, which resulted in its HMG elation. And using this system, they investigated how the HMG elation of fatty acid synthase was influenced by other metabolites. And they found that the HMG signal could be lowered by adding other metabolites. And malonyl-CoA was used, which we know is a chemical substrate of fatty acid synthase. As we can see over here, we know this is what I was talking about earlier about malonyl-CoA coming into play. And we're going to go into more detail here. And so they added HMG-CoA concentrations to fatty acid synthase with various malonyl-CoA also being added. And what they found was as the concentration of malonyl-CoA increased, HMG elation of fatty acid synthase decreased. So they determined that malonyl-CoA competes with the ability for HMG-CoA to modify fatty acid synthase. And they also found that statin-induced HMG elation occurs rapidly, and when statin treatment was removed, the modification disappeared. All of this indicates this modification as being highly dynamic, and they wanted to characterize the bonds formed between HMG and fatty acid synthase after discovering HMG might be bound to fatty acid synthase through amino acids other than lysine. And so they found that the modification is comprised mostly of diester bonds and possible ester bonds, and that is where this next slide is going to lead into. Okay, so now they wanted to identify the exact sites of HMG elation. They were interested in the sites that would form the bonds talked about in the last slide. They were able to identify the prosthetic group phosphopantethine as being HMG elated via a diester bond. And like I said, we talked about phosphopantethine and where it came from, and I believe it was one of the very first slides we talked about, and I just wanted to put the image in your head so now you can know what I'm talking about here and where this is being affected. And so this prosthetic group is directly involved in the catalysis of fatty acids. And additionally, they found serine 581 and lysine 673 to also be a site where HMG elation occurs. The lysine is not of interest for this experiment because lysine modifications require an enzyme to break the stable bond, while the ester and thiester bonds are less stable and do not need an enzyme. And an example that shows this is the malonyl-CoA removing the HMG elation due to the thioester bonds, which we talked about with the, I believe it was maybe 2 or 1 slide before this one. So because of the discovery of multiple sites being affected, they wanted to identify the impact on the activity of fatty acid synthase. For this experiment, they found that the addition of HMG-CoA led to a reduction in activity of fatty acid synthase. They next tested if the fatty acid synthesis of palmitate was impacted in vivo, and they found that it was not impacted. They then tested lipid biosynthesis in a mouse model and found it was not affected despite reductions in fatty acid synthase activities. So, why the conflicting data between in vivo and in vitro experiments? Well, they concluded that the HMG elation seen comes from a localized source of fatty acid synthase that doesn't contribute to the production of palmitate. So, there must be a subpool of modified fatty acid synthase near the site of inhibition at HMG-CoA reductase embedded in the ER. So, as you know, the HMG-CoA reductase is embedded in the ER, and they think that a fatty acid synthase, a subpool of it, is located near it. And this is for the reason why there's the conflicting data between the in vivo and in vitro experiments. And so they performed an experiment to see if fatty acid synthase was directly interacting with HMG-CoA reductase, and they found that yes, they were. So because of this discovery, they wanted to see if there were any cellular changes that occur due to the HMG elation, and they found that several pathway changes overlap in statin-treated cells and inversely change in hypoglycin-treated cells. And in order to determine these pathway changes, they used proteins, specific proteins, and these pathways that were specific to each treatment. And because of this, these pathways imply a change in cellular signaling events centered around the ER and Golgi. And most pathways that were decreased were related to ER and Golgi transport. So finally, they wanted to see if this system was because of statins or other things that could cause this. They used AMPK because it inhibits HMG-CoA reductase, and we have just recently learned about AMPK. And we know that it is activated during times of stress, as well as the inhibition of several enzymes. When conducting the same experiments with AMPK, they found that it did the same as statin-treated cells. They also found that AMPK also affected the same type of pathways connected to membrane and vesicle transport. With this, they concluded that the HMG elation of fatty acid synthase could be driving a change in ER and Golgi membrane transport. This is my first question, and it is, why would HMG elated lysine not be looked at for this study? Second question is, what makes the unusual modification of HMG elevation highly dynamic? There are two explanations, and you can give me one. Thank you.